Good evening, everyone. Whether I'm audible? Good evening. Sorry for the delay. Good evening. Kindly let me know whether I'm audible or not. Kindly drop a message whether I'm audible or not. Yes, sir. Yes. Yeah. Yes. Today we are going to discuss about basic biochemistry. Okay. This is one of the most important topic. Okay. Basic biochemistry followed by we are going to discuss about enzymology. Okay. In basic biochemistry, first we will discuss about the proteins. Okay. Amino acid proteins. Okay. What are the different primary and secondary structures, quaternary structures, how a protein will form. Okay. We are going to discuss everything about proteins. Okay. Followed by we are going to discuss about carbohydrates. What are the basics about carbohydrate? Okay. Then we are going to discuss about nucleic acid. At last we are going to discuss about enzymology. Okay. This is very important lecture. Okay. Very important lecture. These slides are highly sufficient to cover the basic biochemistry syllabus. Okay. So kindly be attentive. If you have any doubt, you can let me know there itself. Okay. First we will start with proteins. Okay. We know the proteins are what? They are polymers of amino acid. This is the general definition. Proteins are polymers of amino acid. Okay. First we are going to see what is mean by amino acids. Okay. What is mean by amino acids? Okay. Amino acids are organic compounds with carbon, hydrogen, oxygen and nitrogen. If you see the basic structure of amino acid, it will have C alpha carbon attached with amino group, carboxy group and then single hydrogen molecule and then R group will be there. Okay. Based on this R group, there are different classifications of amino acids are there. Okay. That we are going to see in our upcoming slide. Okay. So, this amino acid will be polymerized, that will be polymer to form the proteins. Okay. So, proteins are basically polymers of amino acids. Okay. Polymers of amino acids. The individual amino acids in an proteins will be attached by which bond? That is peptide bond. Using peptide bond, the amino acid will be attached together. Okay. This peptide bond is a class of covalent bond. Okay. It's a covalent bond. That means it is very strongest bond. Okay. Strongest bond. Okay. Then the most important thing, while condensing the amino acid with another amino acid, for example, an amino acid is condensing with another amino acid, a molecule of water will be released. This is JRF question. They have asked this one. Okay. So, the amino acid will be condensed. During that process, a molecule of water will be released. This is another important point you have to remember. Okay. There are 20 different standard amino acids are there. Okay. 20 standard amino acids are there. Okay. 20 standard amino acids there. Okay. In some organism, there are additionally 2 standard amino acids are there. Okay. What are that amino acid and what are the codons will code that amino acid? These are the key important thing you have to remember. Okay. If you see the 21st amino acid, 21st standard amino acid is selenocysteine. Okay. Selenocysteine. This is very important, selenocysteine. And 22nd amino acid is what? That is pyrrolysine. Okay, pyrrolysine. Okay, these two are very important. Then the most important thing, which codon will code this selenocysteine and pyrrolysine? Okay, UGA will code for selenocysteine, UAG will code for pyrrolysine. This also important point. Okay, this is very important. Okay, selenocysteine will be coded by UGA, pyrrolysine will be coded by UAG. Okay, I hope this is clear for everyone. Okay, now we are going to see how the amino acids are named. Okay, we know there are different amino acids are there, glycine, alanine, valine, leucine, like that. On what basis they have named the amino acid? If you see, the trivial and common name of the amino acids was given based on the source from where they have isolated, based on source. For example, asparagine was isolated from asparagus. Okay. Glutamate from wheat gluten. This is very important. Okay. Wheat gluten will have predominance of glutamate amino acid. Okay. Tyrosine from cheese. Okay. Glycine means its sweet will be, it will be sweet in taste. That's why they have given the name glycine. Okay. Then another thing I am going to discuss here, which is the first discovered amino acid and which is the last discovered amino acid. This they asked in SRF question. Okay. They asked in SRF 2023. Okay. SRF 2023 they have asked this one. Okay. So you have to just remember NTA. Okay. NTA. A is for amino acid. Okay. So N is the single letter codon for what? For asparagine. Okay. Asparagine. Okay. So first discovered amino acid is asparagine and last discovered amino acid is 309, that is T. Okay. So you just have to remember NTA. First discovered amino acid is asparagine, last discovered is 309. I hope this is clear. Okay. Now we are going to see the general structure of amino acid as we already discussed, that will have an alpha carbon atom bonded with carboxy group and amino group will be there, and H will be common for everything. Based on the R group, they have classified a different amino acids. Okay. This is general structure of amino acid. Okay. And the most important thing you want to say, we want to discuss is that each amino acid will be denoted by a short letter things. Okay. For example, 3-letter abbreviation we can use for denoting each and every amino acid. For example, GLY means glycine. Okay. Or else in bioinformatics, if you see in bioinformatics, large amount of data has to be stored. Okay. For that cases, this 3-letter code was further simplified into a single letter code. Okay. Single letter code. Okay. For example, each and every amino acid was written or was named as single letter codons. This is also another important point you have to remember. Okay. This is just the basic structure. C alpha carbon will be there with attached with amino group and carboxy group. Based on the R group, they are classified into different amino acids. Okay. This is very simple thing. Okay. This is very, very important. From here they will ask match the following question. They will ask match the following question. Okay. Alanine means what? Single letter codon. A. Arginine R. Okay. The difference you have to, what are the different things? For example, G means glycine, that we know. But see here, N. N means asparagine. Okay. D means what? Aspartic acid and glutamic acid. Like that, different things are there. From here, sure, they are going to ask one question. You have to remember properly. For example, tryptophan. Tryptophan means W. Okay. Tryptophan means W. Phenylalanine. Phenylalanine means what? F. Like that, different things are there. You have to remember. Okay. These are the important things you have to remember from basics of amino acid. Okay. Now we are going to see the chirality of amino acids. Okay. Chirality of amino acid. From here, they have asked one time question. Okay. So chirality means what? The anomeric carbon will be attached with 4 different groups. Okay. This is known as chirality. If you see the structure of amino acid, out of 20 amino acid, 19 amino acid will have chiral carbons except one. What is that? Means glycine. Because if you see in glycine, there will be 2 hydrogen atom attached to the C alpha carbon. I hope this is clear for everyone. Okay, so C alpha will be attached with 2 molecule of hydrogen. So glycine is not a chiral molecule. Clear? Glycine is not a chiral amino acid. And one more question what they will ask means, what are the amino acids that is having 2 number of chiral carbons? Okay, you have to just remember IT. Okay. Isoleucine and threonine. Isoleucine and threonine will have 2 chiral carbons. I hope this is clear for everyone. Okay. Isoleucine and threonine will have 2 chiral carbons. Okay. Now we are going to see the classification of amino acid. How the amino acids are classified based on R group. Okay. Before going to that one, I just want to say one simple concept. Okay. The amino acid present in the proteins will be always in L form. Okay. L amino acid will be present in protein. But the D amino acid will be present in cell wall of bacteria. I hope this is clear. In humans and plants, whatever the amino acid present in proteins, that will be present in L form. Okay, L form. So the D amino acid will be primarily present in cell wall of the bacteria. Okay, this is another important point you have to remember. Okay, now we are going to see the classification of amino acid. How the amino acids are classified based on R group. Okay, the first classification is based on R group. They have classified aliphatic R groups are there. For example, amino acids that are non-polar and aliphatic R groups are there. Okay. The primary example of glycine, alanine, proline, valine, leucine, isoleucine and methionine. This 7 amino acid will comes under the class of non-polar aliphatic R group. This is very important. Non-polar aliphatic R group have 7 amino acid. If you see the speciality of each and every amino acid, this we have already discussed. Glycine is a non-chiral amino acid. Okay. Then the most important thing you have to take from this slide is what are the example of branched chain amino acid. Okay. So the example of branched chain amino acid is valine, leucine and isoleucine. These 3 amino acid will have branched chain amino acid. Okay. And the another most important thing, amino acid having imino group. Okay. Not amino. Imino group will be there in proline. Okay. Imino group will be there in proline. And methionine is the example of what? Sulfur containing amino acid. Okay. Sulfur containing amino acid. Now we are going to see the second classification. Okay. Earlier we have discussed non-polar aliphatic group. Okay. Now we are going to see polar with uncharged R group. This is very important. Polar with uncharged R group. The example are what? Serine, threonine, cysteine, asparagine and glutamine. Okay. Serine, threonine, cysteine, asparagine and glutamine. Here, cysteine is another example of sulfur containing amino acid. So, methionine and cysteine is the example of sulfur containing amino acid. Okay. The hydroxy containing amino acid example is what? Serine and threonine are hydroxy containing amino acid. That's why the phosphorylation will always happen majorly at serine and threonine residue of protein. Okay. Then amide containing amino acid, amide, AMIDE, amide containing amino acid is asparagine and glutamine. You see here, amide is there in that R group. Okay. I hope this is clear. Okay. Now we are going to see polar charged R group. Okay. Earlier we have seen uncharged. Now we are going to see charged. We can broadly classify it into positive charge and negative charge amino acid. Okay. The example of positive charge amino acid is arginine, lysine and histidine. These 3 are very, very important. Arginine, lysine and histidine are example of positively charged R group. Okay. And most important thing, this we can also call as basic amino acid. Okay, basic amino acid. Okay. Then what is meant by acidic amino acid? You see here, aspartic acid and glutamic acid is example of acidic amino acid. Okay, that will have negative charge in its R group. This is also another important thing you have to remember. Okay. Now we are coming to the last classification. Okay. Last classification amino acid is aromatic amino acid. Okay. Aromatic amino acid. If you see the example of aromatic R group, phenylalanine, tyrosine and tryptophan are example of amino acid that containing aromatic groups. Okay, for example, phenylalanine will have benzene ring. If this benzene ring is having hydroxy group, that is known as tyrosine. Then next important thing is tryptophan. Here what question they are repeatedly asking means the proteins will have peak absorption at which nanometer of UV range. Okay, you have to remember, okay, at 260 nanometer, okay, 260 nanometer DNA absorption will be there max. 280 nanometer protein absorption maximum will be there, protein. This absorption is primarily because of the presence of aromatic compounds in the amino acid. Okay. Among this aromatic containing R group, tryptophan will have higher absorption. This is also its important point you have to remember. Okay. Tryptophan will have higher absorption. Okay. I hope there is no doubt in classification of amino acid. Okay. Any doubts are there in classification of amino acid based on R groups? Any doubts? Kindly reply any doubts. Hmm? If no other doubts means I will proceed. Okay. Kindly reply any doubts. Yeah, if no other doubt means I will proceed. Still now we have seen what are the example of standard amino acid. Okay, now we are going to see examples of non-standard amino acid. Okay, non-standard amino acid. Okay, from here we can expect question. Okay, the example of non-standard amino acid is hydroxyproline, hydroxylysine and methyl lysine. This is very important because hydroxyproline and hydroxylysine will be present in collagen molecule. This is very, very important question. Okay, that will be present in collagen molecule. Okay, then another most important thing, gamma carboxyglutamate. Okay, gamma carboxyglutamate will be primarily present in vitamin K. This is also another important thing. Next, desmosine. Okay, desmosine, if you see here, this will have a lysine amino acid will be there in it. Okay. So, tetralysine molecule will be there in desmosine. This they have asked one time. Okay. Lysine will be present in desmosine, and another most important thing, this desmosine will be predominant in elastin protein. Okay. This also you have to keep in remember. Elastin protein will have desmosine. Okay. Next, now we are going to see the standard and non-standard amino acid we have completed. Okay, just a minute students, sorry for the disturbance. Yeah. Standard and non-standard amino acid we have completed. From here, sure we will get one or two questions. Okay, amino acid basics you will get minimum one or two questions you will get. Okay, so don't miss even a single point. Okay, now we are going to see the classification of amino acid, whether it is an essential or non-essential amino acid. Okay, so essential amino acid means what? The body cannot able to synthesize that amino acid. Okay, so non-essential amino acid the human body can synthesize. Okay, but in plants all the amino acid will be synthesized. The 20 standard amino acid will be synthesized in plant. Humans cannot able to synthesize this essential amino acid. Okay, this is very important. From here sure you will get a question. Okay, the example of essential amino acid, you just remember aromatic amino acid first. Phenylalanine and tryptophan is a critical example. Apart from that, branched chain amino acid, I already told, valine, leucine, isoleucine, these three are highly important. Then threonine, lysine, methionine and histidine. Total 9 essential amino acids are there. This is very important. If you remember this 9 essential amino acids, remaining things we can solve. Okay. So from this slide, sure you will get 10 question. What are the example of essential and non-essential amino acids? Okay. Now we are going to see the titration curve. Why we are discussing this titration curve? Okay. From this they will ask problems. Okay. How to calculate the isoelectric point of an amino acid, or how to calculate the isoelectric point of a protein. Okay. I hope today also they have asked the same question. Okay. What is mean by isoelectric point? Okay. For this purpose, we are going to discuss about titration curve. Okay. You just remember during our school days, we will do titration. Okay. We will do titration against a standard base. To know the acid concentration, we will take acid in conical flask. Sodium hydroxide will be loaded in that burette. Then we will do titration. Okay. So titration why we are doing means to know the concentration of acid. Concentration of acid we can know. Okay. Every acid will have an typical titration curve will be there. For example, if you see acetic acid, the acetic acid will have a titration curve like this. Okay. For every acid, there will be a particular pKa values will be there. Okay. pKa values will be there. This is another important question. The pKa value for acetic acid is 4.76. This is another important question. Okay. Now we are coming to our actual point. If you see the amino acid basic structure, this is having a carboxy group is there and it is having an amino group. Okay. At a neutral pH, we are saying this will have both positive and negative charge. That means it is a neutral form. Okay. If this amino acid is going to an acidic pH, if the amino acid is going to acidic pH, this H plus concentration will make the negative charge as neutral. Clear? So the negative charge is neutral. So the overall charge on the amino acid will become +1. I hope this is clear for everyone. +1 will be there. If we keep this amino acid in high pH, that is basic pH, that will have a charge of -1. Okay, this is very important. So at neutral pH, the charge will be 0. Okay, at acidic pH, the charge will be +1, and at basic pH, the charge will be -1. We have seen different amino acids are there. Charged amino acid, okay, positively charged, negative charge, like that, different classifications are there. If we want to know isoelectric point of particular protein, for example, I have one protein. Okay, that protein have 10 amino acids. I want to know charge of this protein. Okay, charge of this protein means I have to know at which environment the protein is there. Then I have to calculate based on R group, how the charge intensity of the protein is varying. Okay. Now simple thing, what question they will ask? I will say here itself. Okay. First, what is mean by isoelectric point? This is very important definition. Okay. Isoelectric point means the pH at which the charge of the protein will become neutral. Okay. So isoelectric point means what? pH at which the net electric charge in the protein is 0. Okay. This is very important definition. Okay. If I want to find the isoelectric point, that means pI for a protein. Okay. Just I have to calculate the pKa1 plus pKa2 by 2. That means what? pKa1, that pKa1 will gives the charge of the acidic group, COH group, that it will give. Okay. Then pKa2, that will give the pKa value for amino group. For example, just you imagine, this is the glycine molecule. One glycine molecule is there. They will give the values for pKa1 and pKa2. Okay, for example, pKa1 for glycine is 2.34 and pKa2 is 9.30. If they given like this and to calculate, and they calculate the PI, means you have to just take the mean of these two value. The formula for calculating PI is pKa1 plus pKa2 divided by 2. If you see here, pKa1 is 2.34, pKa2 is 9.60. So if we add and divide by 2, the value will be 5.96. So 5.96 is the PI for this glycine. I hope this is clear. Okay. So if you see the glycine PI, the value is 5.97, and the formula for calculating the PI is what? pKa1 plus pKa2 divided by 2. I hope this is clear for everyone. Clear? 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 Yes. If clear means we are proceeding next thing. Okay. Now we are coming to little much complicated things. How means? We know glycine will have only H plus molecule. If you see some of the other amino acid like glutamic acid, that will also have the R group. That R group will have the negative charge. So that may influence the PI. Okay. So in that case, the formula of PI will come pKa of acidic group plus pKa of R group by 2. Okay, you just imagine. Okay, I will say one thing. Okay, if monoamino monocarboxylic acid means PA calculation formula is pKa1 plus pKa2 divided by 2. If diamino monocarboxylic acid means pKa of 1 plus pKa of R by 2. I will explain, don't worry, okay? Then monoamino dicarboxylic acid means pKa2 plus pKa by 2. This is the formula you have to remember. Okay? For example, if you see glycine, that will have C alpha carbon, carboxy group will be there, amino group will be there, H and H will be there. Okay? Here only one amino group is there and only one carboxy group is there. So this is the example of monoamino monocarboxylic acid. So that time we just have to calculate the pKa for this and this divided by 2. Clear? If it is a dicarboxylic acid, that means what? If you see glutamic acid, it will have R group. This R group also will have COH group. In that case, we have to calculate pKa1 plus pKr. Okay, I hope this is clear. From here, sure you will get problem questions. You will get. Okay. Okay, this is for histidine charge group they have given. Okay, I hope the basics of amino acid is clear for everyone. Any doubt is there? Kindly let me know any doubt is there. Lokendra sir paper बहुत अच्छा. Okay, very good. Translation question नहीं हुआ. Translation is very important. Translation inhibitors are very important. They will ask in main paper. Don't worry. Okay. Kindly let me know any doubts are there. I need interaction session. Any doubts? Any doubts? If no other doubts means we can proceed. Okay, next thing. Okay. So far we have discussed A to Z of amino acid. Okay. Now we are going to see how these amino acids are condensed to form protein. Okay. So protein means what? Proteins are most abundant biological macromolecules occurs in all cells. Okay. This is what? It's an polymer of amino acid. Proteins are polymer of amino acids. They are joined by peptide bond. Okay. Peptide bond is an example of what? It's an example of covalent bond. Okay. Or it's a type of amide bond. These all are interchangeable. Okay. So it's an example of amide bond. Okay. Another most important thing we are going to discuss now. Okay. I already told the individual amino acid. This is amino acid 1. This is amino acid 2. The N-terminal of the amino acid will join with C-terminal. That means carboxy group will bind with amino group. Okay. This is a condensation process, that is because of condensation a molecule of water will be released that lead to the formation of this peptide bond. I hope this is clear. Okay. If you see the peptide bond, C double bond O NH will be there. This is peptide bond, and the most important thing what they will ask means this peptide bond is what? It's a partially double bond nature will be there. Partial double bond nature will be there. TK double bond nature will be there. And another most important thing, the peptide bond will be always present in trans form. You see, the carboxy oxygen and amino hydrogen will be always presented in opposite side. So peptide bond is a trans bond. TK. But if you see another most important thing, in lipids, the double bond will be present in cis form. Okay, lipids or fatty acid, the double bond will be present in cis form. This also you have to remember. Okay. Then another most important thing is just a simple term. Okay. What is meant by dipeptide? Dipeptide means two amino acid will bind to form a single peptide bond. I hope this is clear. So dipeptide means two amino acid will be there, but only one peptide bond will be there. Tripeptide means three amino acid will be there and two peptide bond will be there. This is just for your understanding. This question they can ask in assertion and reason. Okay. This is assertion and reason question they can ask. Okay. So this is very important. Okay. Now we are going to see the characteristics of peptide bond. We already discussed peptide bond. It's a rigid bond and it is trans in nature. Partial double bond nature will be there. Okay. And another most important thing what I have to discuss here, if you see here, this is C alpha carbon. Okay. This is also C alpha carbon. Okay, this is peptide bond. There is a possibility of rotation of this C alpha and C bond. This is clear. C alpha and C bond can rotate here. Okay. This CN is what? It's a double bond nature. So rotation will be restricted almost. It will be restricted. But if you see here, this N and C alpha bond, N and C alpha bond also can rotate. Okay. This can rotate with 360 degrees. 360 degree it can rotate. Okay. Now, based on this rotation, the which angle should be there. Okay, for example, if you see there is an arc, there is arc groups in each and every amino acid. So, the particular rotation of this C alpha and C bond and N C alpha only will determines the secondary structure of protein. If the bond angle is not correct, that may be sterically hindered. That means the bonds will not form properly. Okay. This is very important. Why I am saying means this rotation, as Ramachandran, like that one person is there, what he did means he has detected what are the particular C alpha and C bond and N and C alpha values that is possible to determine the secondary structure of protein. Okay. I hope this is clear. Okay. This is clear. Okay. Now we are going to see if the C alpha C bond is rotated, C alpha C bond is rotating, that name is known as psi bond. This is very, very important. Okay. C alpha C means psi bond, N C alpha means pi bond. Okay. This is very, very important. C alpha C means what? N C alpha means what? This is very, very important. That's why we are called as dihedral angle. Okay. You just see here. C alpha C means psi, N C alpha means pi bond. Okay. This is very important. Again, I am saying this is very important. Okay. Ramachandran plot we have told, okay, what are the possible rotations that will create the stable secondary structure of protein was elucidated by Ramachandran and the plot was given like this. Okay. See here, pi values are there, psi values are there, total 360 degrees there. Okay. So wherever the possible regions are there, so in this region proteins will form, protein secondary structure will form. Here also protein secondary structure will form. Like this, he has detected the pi and psi values. Okay. Here what question they will ask means, here what question? Glycine is less sterically inhibited. Okay, less sterically hindered. Why means it will have only H group as an R. Only H will act as an R group of this glycine. So it is less sterically hindered. Okay, glycine is less sterically hindered. This is very important thing. Okay, you have to remember. Okay, coming to the next question. Okay, now we are going to see the structures of protein. From here, in 2024, they asked two questions. Okay, from structures of protein. Okay, so this is very important topic. Okay, there are 4 different structural classifications of protein are there. Again, I am saying structural classification of proteins are 4 types. Okay, first is primary structure, secondary structure, tertiary and quaternary structure. This figure is highly sufficient. You see here, primary structure means it's sequence of amino acid in a protein. Okay. Primary means what? Sequence of amino acid in a protein. So secondary structure means what? This amino acid will attach, this amino acid will form a bond with further away from the amino acid. For example, in alpha helix, N and N plus 4th amino acid will form a bond. Okay. N and N plus 4th amino acid will form a bond. This is known as secondary structure. There are different classes of secondary structures are there. Like alpha helix will be there, beta sheets will be there, like that. Different secondary structures are there. That we are going to discuss now. Okay. So tertiary structure means what? It's a 3-dimensional structure. Okay. It's a 3-dimensional structure of protein is known as tertiary structure. Okay. So what is meant by quaternary structure? If a 3-dimensional structure, one polypeptide bind with 3-dimensional structure of another polypeptide. Okay, so multiple 3-dimensional structure, or we can repeatedly we can say a polypeptide. Okay, a protein made up of more than one polypeptides are known as quaternary structure. I hope this is clear. You see here one protein is there, take a polypeptide, here one polypeptide is there, then here also different polypeptide, here also different polypeptide. Alpha 2 beta 2 structure is there here. Okay, this is known as quaternary structure. Okay, just we are going to see the definitions now. Okay, so primary structure means what? It's the sequence of amino acid, and most important thing, primary sequence that will determines the secondary and native structure of protein. Okay, so the amino acid sequence will determine the native secondary structure and tertiary structure of the protein. This is another important point you have to remember. Okay, secondary structure, this we already discussed. Okay, it is referred to particular stable arrangement of amino acid residues. Okay, particular stable arrangement of amino acid residue, and most important thing, secondary structure are highly stabilized by hydrogen bonds. This is also another important thing you have to remember. Okay, hydrogen bonds. Okay, now we are going to see what are the different classes of secondary structure. Okay, simple thing, the major secondary structure is alpha helical form. Okay, then beta pleated sheet. Okay, beta pleated sheet. You just remember these are the two major secondary form. Okay, in alpha helix means N and N plus fourth amino acid will join. This is JRF 2024 question. Okay, so N and N plus fourth amino acid will form bond in alpha helix. Okay, in beta pleated sheet there are two different types are there that we are going to see here. Okay, so parallel beta sheet will be there and anti-parallel beta sheets will be there. Okay, so two different forms of beta sheet will be there. Among them, anti-parallel beta sheets are highly stable. Anti-parallel beta sheets are highly stable. These are the key points they will ask in exam. Okay, this is just a structure of alpha helix. I already told N and N plus fourth amino acid will join. Okay, N and N plus fourth amino acid will join. Okay, the helix length of helix length, if you see, it will be around 3.6, sorry, 3.6 amino acid will be there per alpha helix, and if you see the width of this helix, that will be around 5.4 Armstrong. This is another important question. Okay, so 5.4 Armstrong will be there, this height or height of this alpha helix. Okay, in this total 3.6 amino acids will be there. These two are very important thing. The alpha helix we can also called as 3.613 helix. This is also another important point. Alpha helix we can also called as 3.613 helix. I hope this is clear for everyone. Okay. Now secondary structure, beta pleated sheet, this we have already discussed. Okay. In beta sheet, the most important thing what they will ask means anti-parallel beta sheets are highly stable as compared to parallel beta sheet. This is the key point you have to remember in beta sheets. Okay, clear? This is Ramachandran plot. We have already discussed this one. Okay. From this Ramachandran plot, what question they will ask means, what is the pi and psi value for alpha helix structure? Okay. If alpha helical structure has to form, the pi angle should be minus 57 degree and psi angle should be minus 47. Okay. This is very important question. Okay. Pi and psi value. You just have to remember the pi and psi value for these two things. Okay. Alpha helix and beta parallel sheets. Okay, parallel and anti-parallel sheet. From here, they will ask question. Okay. Now we are going to see how we can determine the secondary structure of protein. Okay, which technique we can use for determining the secondary structure of protein. Okay, we can use circular dichorism. This is a technique. This will be used to determine particular secondary structure in a protein, whether it's having alpha helix or beta sheets or turns, whatever it will be there in the protein, that we can determine by circular dichorism. Again, I am saying this is very important point you have to remember. Okay, you see here this figure. Okay, based on the change in ellipticity, we can determine the secondary structure of proteins, whether it is having alpha helix or beta sheets or random coils are there. that we can determine. I hope there is no doubt in secondary structure of protein. Major is alpha helix and beta pleated sheets. Okay. Now we are going to see about turns. Okay. You just imagine this is one alpha helix, this is another alpha helix. These two alpha helix has to be joined by what? By turns, by loops and turns. Loops and turns are highly useful for joining the secondary structure of proteins. Okay, this is important. Okay, you just have to remember the definition. Okay, loops and turns will be used for joining the secondary structures of protein. Okay, here there are different classifications are there. Just you go through once. Alpha turn, gamma turn, and beta turn, like that is there. And the most repeated question what they are asking means glycine and prolines will be predominant in turns. Glycine and proline amino acids will be predominant in turns. Why glycine and proline is predominant? If you see, we want to make a kink or we want to just make a bend in that protein. Okay, so if you see, glycine is what? It is having only H group, that is easy flexible. But proline, if you see, imino group will be there, that is sterically hindered. So it will be easy to make a flexible structure in the proteins. Okay, that's why the glycine and proline are highly predominant in turns. Okay, then tertiary structure, this is another important thing we have already discussed. It's a three-dimensional conformation of a protein. Okay, this will be highly stabilized by hydrogen bond, hydrophobic bond, and weak forces also will be involved in stabilizing the tertiary structure. Okay, apart from that, covalent bond will also involved in forming the tertiary structure. How means? You just imagine, okay, cysteine will bind with another cysteine through disulfide bond. Okay, disulfide bond will be formed. Okay, this is an example of covalent bond. Okay, and last we are going to see about quaternary structure. This is also we have discussed. Okay, quaternary structure means two or more polypeptides can be joined together to form the proteins. Okay, two or more proteins can be joined, two or more polypeptides can be joined to form the protein. I hope this is clear for everyone. Okay. Any doubt is there in structural classification of protein? Kindly reply, any doubt is there in structural classification of protein? If no other doubt means I will proceed other classification of protein. Just 10 minutes we will complete the proteins and we can have a break. Any doubt? Any doubt? If no other doubt means I will see the next classification of protein. Yes. Yes. Okay. The proteins can also be classified as three different things like simple protein, conjugated protein, and derived protein. Okay. These are the other examples. Okay. Simple protein means it will be primarily made up of only proteins. Okay. Composed protein means it will have other than the protein molecules will be there. Other than amino acid, for example, ions will be there. For example, iron, magnesium, like that will be there. Things will be there. Okay. So that are known as conjugated proteins. Okay. You just go through once this one. This is not that much important, but just go through once. Okay, what are the different classes of protein? Lipoprotein, glycoprotein, like that. Different classifications are there. Among them, what are the examples and prosthetic group? You just go through once. Okay, lipoprotein, glycoprotein, phosphoprotein, hemoprotein, flavo and metalloprotein. These are the different examples or different classes. Okay. Then what are the prosthetic group present in that and what are the examples? You just go through once. Okay. Now we are going to see, still now we have seen how the proteins are formed. Okay. Now we are going to see how the proteins are proteolized. That means how the proteins are degraded. Okay. From here, 2023 and 2024 continuously they have asked the same question. Okay. So if you see ubiquitin mediated proteolysis, what is mean by ubiquitin and what is mean by ubiquitin mediated proteolysis? Okay. This ubiquitin is itself a protein. It's an 76 amino acid protein. Okay. What it will do means if the proteins are tagged with ubiquitin molecule, that will be targeted for proteolysis. I hope this is clear. Okay. If a protein is there, if that is tagged with ubiquitin molecule, that will be entered to proteolysis. That means it will enter to degradation process. Okay. So where this ubiquitin molecule will tagged in the target protein, if you see the lysine residue of the target protein, in the lysine residue of the target protein, this ubiquitin molecule will be attached. I hope this is clear for everyone. Okay. So lysine amino acid, it is the target wherever ubiquitin molecule will attach. Okay. There are three different enzymes are involved in the protein mediated, involved in proteasomal mediated degradation. Okay. Three enzyme, enzyme 1, enzyme 2 and enzyme 3. In 2024, what they asked means what are the different enzyme? They have given the name. What is enzyme 1, what is E2 and what is E3? This they asked. Okay, you just remember. Okay, E1, E1 is ubiquitin activating enzyme. Then E2 means ubiquitin conjugating enzyme. E3 means ubiquitin protein ligase enzyme. Okay, this is very important. I am saying again. U1 is ubiquitin activating enzyme. U2 is ubiquitin conjugating enzyme. And U3 is what? Ubiquitin protein ligase. This is very important. Ubiquitin protein ligase. This is the three classification or three enzymes involved in ubiquitin mediated protein degradation. Okay. How it will be degraded means after tagging the proteins by ubiquitin, it will be entered to 26S proteasome. Okay. 26S proteasome. It is nothing but it's like a barrel-like structure will be there. The protein will enter inside. There will be protein degrading enzymes will be there that will cleave the entire protein into separate amino acids. Okay, I hope the concept is clear. Whatever questions they will ask, that also we have discussed. Okay, now we are going to see what are the classification of proteases. This is the last topic, classification of proteases. Okay, if a protein has to be degraded, different proteases should be there. Okay, but each and every proteases will have specific target. Okay, for example, if you see, these are the typical example of proteases. Okay, trypsin, this is the question they are going to ask. Okay, trypsin, the cleavage site will be arginine or lysine, particularly at C terminal of the amino acid. Okay, so C terminal of the protein, lysine and arginine amino acid is the target cleavage specificity for trypsin. And chymotrypsin means aromatic amino acid will be there, particularly at C terminal. Okay. Pepsin means same aromatic amino acid, but it will cleave at N terminal. Okay. I hope this is clear. Okay. Then last class is elastase. The specificity will be alanine, valine, serine and glycine, particularly at C terminal. These three are very important. Trypsin, chymotrypsin and pepsin are very important. Okay. Now we are going to see the last thing. Okay. How we can determine the N terminal and C terminal of an protein. Okay. There are different methods are there. Just you have to remember what are the different methods used for N terminal and C terminal protein determination. Okay. So N terminal protein, N terminal amino acid of a protein can be determined by Sanger's method. Okay. Sanger's method. He got Nobel Prize in 1953 for his discovery. Okay. Here they will use fluorodinitrobenzene they will use. Okay. Fluorodinitrobenzene he will use. That reagent we can call as Sanger reagent. Okay. Sanger reagent means what? 2,4-dinitrobenzene. Okay. 2,4-fluorodinitrobenzene. Okay. Then another most important thing is Dancel chloride method. Okay. Then Edman degradation, mass spectroscopy and aminopeptidase digestion. These are the different methods used for determining the N terminal of an N-terminal amino acid of a protein. Okay. Then another thing, C terminal. How to determine the C terminal of a protein? Okay. The most important thing. This is JRF 2021 question. Okay. In N-terminal determination, we will use hydrazinolysis. Okay. Hydrazinolysis is the major method for determining C-terminal protein. Okay. C-terminal of an protein. Apart from that, carboxypeptidase A and B and carboxypeptidase Y digestion we will use for determining the C terminal of a protein. Okay, I hope there is no doubt in proteins. Any doubt is there? Any doubt is there? With this, we are completing the proteins, amino acids and basic proteins. Here, any doubt is there? Kindly let me know, or else we can have a break for 10 minutes. Sorry, break for 5 minutes. We can resume at 6:25, 6:22. Any doubt? Yes, no doubt means we can resume after 10 minutes. Okay. Shall we start, students? Yes. Now we are going to see the basics of carbohydrates. Okay, we are going to see the basics of carbohydrates. Again, I am saying what are the important things to be asked in the examination point of view. That is the things I am going to discuss. Okay, broadly the carbohydrates are classified into the carbohydrates are classified based on the number of carbons and based on the functional group into different types. Okay. So, if we see based on the functional group, they are classified into aldoses and ketoses. Okay. Aldoses means what? Aldehyde group will be there. Okay. Ketoses means what? Ketone group will be there. Okay. C double bond O. Ketone group will be there. Okay. Aldoses means CHO. That means aldehyde group will be there. Okay. Then based on the number of carbon atoms, they are classified into trioses, tetroses, hexoses, pentoses, like that they have classified. Okay. So trioses means 3 carbon atoms will be there. Tetroses means 4. Pentoses means 5. Hexoses means 6. Okay. Like this they have classified based on the number of carbon atoms. Okay. I hope this is clear for everyone. Okay. From here, what question they will ask means, what are the example of trisaccharide, tetrasaccharide, like that question they may ask. Okay. Next, we are coming to discuss the most important thing, what are mean by epimers. Okay. What are mean by epimers. Okay. So, epimer means what? Two different biomolecules. So, two different molecules that are differ only at the chiral center of single carbon. Okay. For example, these all are chiral carbons of this mannose. These are chiral carbon of glucose. If you see only at the carbon 4, if you see the carbon 4, there is same only it's there, but if you see at carbon 2, see this carbon 2, the arrangement of H and O plus OH was differ. So, the glucose and mannose are epimer at carbon 2. This is repeated question. Glucose and mannose are epimer at carbon 2. Glucose and galactose are epimer at carbon 4. You have to remember the carbons are 2. Carbons also, whether carbon 2 or carbon 4. These two are very important thing. Okay. Now we are going to see the cyclic structure. In first slide, we have seen the linear form of sugars. Okay. Sugars have a capability of forming the cyclic structure. Okay. It may form pyranose structure or furanose structure. You just have to remember furanose. Furanose means 5-member ring structure will be formed. Okay. Pyranose means 6-member ring structure will be formed. So, furanase and pyranose will be there. Okay. So, ring structures will be formed. If a ring structure will be formed, the number of anomeric carbon will increase by 1. This is also another important thing. Okay. Normally, if you see glucose, there is 4 anomeric carbon will be there. If this glucose is found in cyclic structure, the anomeric carbon will become 5. Okay. Anomeric carbon will become 5. So, the formula for determining the number of stereoisomers is 2 power n. Okay. This is also another 2 power n minus 2. This is also another important thing you have to remember. Okay. So, here we have seen what is mean by furanose, what is mean by pyranose. Apart from that, how the cyclic structures are formed, that we have discussed here. Okay. Now we are coming to another important classification. The carbohydrates can be classified into monosaccharides, oligosaccharides and polysaccharides. Okay. Monosaccharide means a single sugar molecules will be there. Oligo means 2 to 10 monosaccharides will be joined. Okay. That is known as oligosaccharides. Polysaccharides means more than 10 monomeric units will be joined through O-glycosidic bond. Okay. These polysaccharides are further classified into 2 types. Okay. This is very important. Okay. They can classify into homopolysaccharides and heteropolysaccharide. Okay. Homopolysaccharide means what? The sugar molecule will be same. For example, if you see in glycogen, okay, if you see the glycogen structure, n molecule of glucose will be repeated there. Okay. If you see in inulin, fructose molecule will be repeated n number of times. Okay. I hope this is clear. That is known as homopolysaccharide. Heteropolysaccharide means more than one monosaccharide units will be there. Okay, more than one different types of monosaccharide units will be there. Okay, these are the two broad classifications of polysaccharides. Okay, then another most important thing, questions what they will ask from basics of carbohydrate means what are the linkage that are present between disaccharides. Okay, for example, different disaccharides will have different linkages. Okay, that is very important question. From there they will ask. Okay, for example, we are seeing here lactose will have beta 1,4 linkage. Okay, so lactose will made up of glucose and galactose. The linkage will be beta 1,4. Then another most important and repeated thing is sucrose. Sucrose is an example of non-reducing sugar. Apart from that, trehalose is also an example of non-reducing sugar. Okay, so two examples are there for non-reducing sugar, sucrose and trehalose. Okay, why they are non-reducing in nature means both the anomeric carbon of the disaccharide will be involved in bond formation. Okay, if you see in sucrose, the linkage will be alpha beta 1 to 2 of glucose and fructose. Okay, but if you see trehalose, the glucose and glucose will be joined together. Okay, glucose and glucose will be joined together by alpha 1 to 1 linkage. Okay, Pranav VJK sir, plant biotechnology का PhD session भी करवाओ please. Yeah, sure, we will start the PhD classes also. Don't worry. Okay, we will sure class for start for PhD also. Okay, so these are the primary important things you have to remember about the basics of carbohydrates. What are the sugars and what are the linkages present in that. Okay, if you see cellulose, the primary linkage will be beta 1,4 will be there, and amylose means what? Alpha 1,4 repeating units will be there, and dextran, this will be present in microorganism. Primarily it will have alpha 1,6 with alpha 1,3 branches. Okay, these are the important points you have to remember. Okay, I already told the important things. This is highly sufficient to cover the basics. Okay, here I have listed what are the different polymers and what is the type and what are the repeating units. Okay, everything we have listed here. Okay, the most important thing, chitin. The linkage in the chitin will be beta 1,4 linkage will be there, and beta 1,4 linkage will be there. This is very important and repeated question. Okay, beta 1,4 linkage will be there. Okay, ज़्यादा रिसोर्स नहीं है यूट्यूब में पीजे ICAR से बेटर, so please. Okay, sure we will start thing. We will start. Okay, next we are going to start nucleic acid. I hope there is no doubt in carbohydrates. Whether any doubts is there on basics of carbohydrates. Carbohydrate metabolism yesterday itself we have covered. Okay, now we are going to see the basics only. Okay, now we are going to see the basics only. Okay, so we are coming to nucleic acid. This is very simple topic. Okay, in nucleic acid, the primary thing in basics what you have to remember means what are the different types of purines and pyrimidine bases. Okay, then second most important thing, classification of aDNA, bDNA and zDNA. Okay, from there they are asking every year question. Okay, these two are the important things you have to learn in nucleic acid. Okay, so if you see purines, the two major example of purines are adenine and guanine. Okay, adenine and guanine. And for pyrimidines, cytosine, thymine and uracil are typical example. Okay, cytosine, thymine and uracil are example of pyrimidines. Okay, then another most important thing, the bases will be joined together by phosphodiester bond. This is also another important thing. Diester bond will be there for joining the bases. Okay, then another important thing, this is JRF 2025 question. Okay, RNA molecules are highly susceptible to alkaline hydrolysis. Why? They have asked this in statement and reason question. Okay, RNA molecules are highly susceptible to alkaline hydrolysis due to the presence of 2' hydroxy group. Okay, at the 2' hydroxy group will be present in the RNA molecule. Okay, but if you see in DNA molecule, deoxy sugars will be there. Because of that, alkaline hydrolysis is not there in DNA molecule. I hope this is clear. Okay, this they asked in assertion and reason. Okay, now we are coming to discuss the actual thing. Okay, What is the key importance between A DNA, B DNA and Z DNA? What are the questions they will ask? We will discuss here. Okay. There are three predominant forms of DNAs are there. Okay. A DNA, B DNA and Z DNA. Okay. A DNA will be highly present in dehydrated condition. Dehydrated condition. Okay. B DNA will form in normal condition. Okay. Normal cellular condition will have B form. Okay. Z DNA will be present in high salt condition. Okay. These 3 points are very important. Okay. So A DNA will be predominant in dehydrated condition. B DNA will be found in normal condition and ZDNA will be predominant in high salt condition. Okay. Then another most repeated thing what they are asking means left-handed alpha helix will left-handed helical form will be predominant in Z form. Okay. Z means left-handed helical will be there. Okay. These two things are very very important. If they want to make question paper tough, they can ask the sugar puckering. Okay, how the sugar puckering is there. Okay, you just have to remember in BDNA the sugar pucker will be C2 endo. Okay, C2 endo. Okay, C2 endo. In ZDNA, pyrimidines will have C2 endo and purines will have C3 endo. Okay, these things are very important. And in A form, the sugar will be always puckered in C3 endo form. These three are very important point you have to remember. At last, just you have to remember the number of base pairs per helical turn. Okay. A DNA means what? 11 base pair will be there. B DNA means 10.5 and Z DNA means 12 bases will be there. Okay. 12 base pairs will be there per helical turn. Okay. With this, we have completed the basics of this nucleic acid. That is highly sufficient to cover the basics of nucleic acid. Okay. Now we are going to start. Just a minute. Now we are going to start fatty acids. Okay. Any doubt is there up to this? Proteins, carbohydrates. Any doubts are there? Nucleic acid. Any doubts? Kindly let me know. Kindly let me know any doubts are there. Yes, if no other doubt means I will start fatty acids. Okay. So fatty acids, okay, the name itself it's there. It will have an acid group. Okay. See here, one acid group will be there. Then CH2 single bond CH2 bonds will be there. Okay. So bond form is there. Okay. Another most important thing, methyl group will be there at the end. Okay. So how we are naming the fatty acids? Okay. Fatty acids, na? There are two ends are there. Okay. Omega carbon is there, then alpha carbon is there. Okay. Omega carbon means what? The carbon atom that is furthest away from the methyl group. Okay. The carbon atom that is further away. Okay. This is very important. And most important and repeated question what they are asking means what are the example of omega-3 fatty acids? Okay. This is the repeated question. EPA. EPA means what? Eicosapentaenoic acid. This is a typical example for omega-3 fatty acid. Okay, omega-3 fatty acid. Then another example you see, this is example of omega-9 fatty acid. Okay, omega-9 is what? Linolenic acid. Okay, linolenic acid is example of, oleic acid is example of omega-9 fatty acid. Okay, oleic acid is example of omega-9 fatty acid. You have to, from fatty acid, what question they will ask means, what is the number of carbon atom for fatty acid? For example, they can give stearic acid. Stearic acid means what is the number of carbon atom? 18 carbon atoms will be there like that. They can ask the number of carbon atoms and they can also ask what are the example of omega-3, omega-6 and omega-7 fatty acid. This we have discussed very deeply in our lecture series. Okay. So just I am giving here for your revision only. Okay. So 12 carbon fatty acid is lauric acid. 14 means myristic. So we have to remember LMP. LMPS. Okay. 12 means lauric, 14 means myristic, 16 means palmitic, 18 means stearic. This is very simple. So 20 carbon is what? Arachidonic acid. This also we have discussed. 20 means arachidonic acid. Okay. So 24 carbon is there means that is lignoceric. Lignoceric means 24 carbon atoms. Okay. Apart from that, we are going to see unsaturated fatty acid. These all are saturated fatty acid without double bonds. Okay. Here now we are going to see the example of unsaturated fatty acid. You just see here 16:1 del 9, that is what? Palmitoleic acid. Okay. 18:1 del 9 is what? Oleic acid. You just have to remember what are the example of unsaturated fatty acid. Okay. Now we are going to see how the melting temperature of saturated fatty acid is different from unsaturated fatty acid. Okay. If you see in saturated fatty acid, there will be an efficient packing will be there in the hydrocarbon chains. Okay. Because of that, the melting temperature will be high. Okay. So saturated fatty acid means melting temperature will be high. Okay. If unsaturated fatty acid means there will be kinks. This double bond will make kinks in the hydrocarbon chain. Because of that, efficient packaging will not be there. So melting temperature will be decreased. These two are very important point. Okay. Melting temperature will be decreased. These two are very important point. Okay. Now we are going to see the classifications of lipid. This we have already discussed in yesterday only. Okay. So storage lipids, the example of triacylglycerol. Apart from that, membrane lipids are there that we have already discussed. Okay. Phospholipids, sphingolipids, everything we have discussed. This also we have already discussed in the yesterday's lecture. Okay. What are the examples of phospholipids? So just I am skipping. Then example of sulfolipids also we have discussed. Okay. Now we are coming to the actual thing. What is the important thing in sphingophospholipid? Okay. So if we see sphingolipids, there are some of the diseases are there. Okay. If the particular enzyme is absent, it will lead to some of the disease in sphingolipids. Okay. That is known as sphingolipid storage diseases. Okay. One time they have asked question from this. Okay. If the deficiency of exosaminidase A enzyme is there means that will lead to Tay-Sachs disease. Only two things you have to remember. Sphingomyelinase enzyme deficiency is there means Niemann-Pick disease will cause. Okay. This you have to just remember only. Okay. Tay-Sachs disease is due to deficiency of exosaminidase and Niemann-Pick is due to deficiency of sphingomyelinase. Okay. Deficiency of sphingomyelinase. Okay. These two are very important point you have to remember. Okay. Now we are coming to the last section of today's lecture, enzymes. Okay. From enzymes, again I am saying you will get 2 to 3 question from enzymology. Okay. You will get 2 to 3 question, at least 2 to 3 questions from enzymology. Okay. We are just going to brush up the concept. Okay. What is enzyme? What are the different classification of enzyme? How we are naming the enzyme? Just we are going to just brush up the concept. Okay. So enzyme means what? It's a biological catalyst that will speed up the essential chemical process in a living organism. Okay. Without being used up. This is very important. I hope today in question paper they ask this question. Okay. Enzymes will be never utilized there. That molecule will not be never utilized. Just it will assist in conversion of substrate to product. Okay. The characteristics of enzyme, if you see, almost all of the enzymes are proteinous in nature except ribosomes. Okay. Except ribozymes. Okay. So, ribozymes are what? They are RNA molecule that is having an catalytic activity. Okay. Then we have given the different special characters. So, enzymes are proteinous in nature, that will be heat labile in nature, then water soluble in nature, they we have given here. Okay. Now we are going to see the brief history. Okay. In this history, the key thing you have to remember is who has given the term enzyme. Kuhne has given the term enzyme. This is very important. Then James Sumner crystallized the enzyme urease. These two are very important point you have to remember. Okay. James Sumner crystallized the urease enzyme. Okay. Now we are going to see the classes of enzyme. Okay. Again I am saying there are 6 classes was there before 2018. Before August 2018, there are 6 major classes of enzyme are there. After August 2017, they have added the 7th class. The 7th class of enzyme is known as translocalase. This is the important thing you have to remember. Okay. So, each enzyme is given or assigned with enzyme commission number. Okay. to enzyme commission number they have given. Okay. Why they have given everything we have discussed in our lecture, if I want to say means, for unifying the names, they have given the enzyme commission number for each and every class of enzyme. Okay. For example, this enzyme commission will have 4 digits that are separated by dots. Okay. The first thing will determines the major class of the enzyme, second will determine the subclass, and third will determine the subgroup, and fourth will determine the number of that particular enzyme on the list. Okay. This is just for your understanding only. So EC will have 4 digits. Okay. Then coming to the classification of enzymes, okay, total 7 major classes are there. From here, what question they will ask means, what are the, if oxidoreductase means which class it will come, class 1, class 2, class 3, like that they can ask. Okay. So oxidoreductase is class 1, transferase class 2, hydrolase class 3. Okay. Then lyases, isomerases, ligases 4, 5 and 6. The new class is what? Translocalase. Okay. Translocalase is the new class. Okay. Next thing, we are going to see the examples. Okay. What is meant by oxidoreductase? Oxidoreductase means oxidation and reduction reaction will be carried out by that special class of enzymes. Okay. Transferase, it will involved in transfer of atoms from one substrate to another. Okay. Then hydrolase, the name itself is there, water molecules will be utilized there. Lyases, that means bond breaking will be there. Isomerases means what? Transfer of group from one position to another. Okay. Then ligases means what? It will joining. It will involve in joining the things. Okay. Then translocalase, that means what? It will catalyze the movement of molecules or ions across the membrane. Okay. It will catalyze the movement of ions or molecule across the membrane. Okay. The typical example is what? ATP synthase complex. ATP synthase complex. Okay. If you see the basics, the enzyme will be made up of proteins. Okay. Apart from that proteins, it will also have non-protein components will be there. Okay. For example, the proteinous component of the enzyme is known as apoenzyme. Okay. Apoenzyme is what? It's the proteinous component of the enzyme. Okay. So holoenzyme means what? The proteinous part along with the non-protein compound. This complex is known as holoenzyme. Okay. Now we are going to discuss what is that non-protein component in an enzyme. Okay. Now we are going to see what is the non-protein component in an enzyme. Okay. You just remember here. Okay. So non-protein component of an enzyme is known as cofactors. Okay. Cofactor. Okay. Non-protein component is cofactor. Now we are going to see what are the classification of cofactor and what is the role of cofactor. Okay. A cofactor is what? It's a non-protein molecule or a metal ion that will increase the catalytic efficiency of an enzyme. Okay. Catalytic efficiency of an enzyme will be increased. Okay. This cofactor can bind either covalently or non-covalently to the enzyme. Okay. Then another most important thing I am going to say here, if the cofactor is binding covalently to the enzyme, that is known as prosthetic group. Okay. So prosthetic group means it will bind covalently to the enzyme. Okay. So then what is meant by coenzyme? Coenzyme means these are organic molecules that can bind loosely to the enzyme. So this is the key difference between cofactor, coenzyme and prosthetic group. Okay. Here they have given the different coenzymes that I have summarized here. From here they will ask the question. Okay. This is very important. Okay. For example, thiamine pyrophosphate will act as a coenzyme for transketolase reaction or transketolase enzyme. Okay. So transketolase enzyme requires thiamine pyrophosphate as a cofactor. Then most repeated question is biotin will act as a cofactor for carboxylase reaction. Okay, I have just given here what are the different coenzyme for different enzyme dependent reactions. Okay, so then another most important thing, tetrahydrofolate will act as a coenzyme for one carbon unit transfer reactions. Okay, tetrahydrofolate. These three are very, very important. Okay. So TPP will act as a cofactor for transketolase. Carboxylation reaction will require biotin as a cofactor. Tetrahydrofolate will act as a cofactor for single carbon transfer reaction. Then another most important thing, what are the metal ions required as a cofactor for enzymes? Okay. From here, sure they will ask one question. Okay. From this coenzyme and cofactor, sure you will get one question. Okay. Urease. This is urease means nickel will act as a cofactor. Okay. Molybdenum containing enzyme is what? Nitrogenase is a molybdenum containing enzyme. Okay. Magnesium will act as a cofactor for hexokinase. Potassium pyruvate kinase. Copper for what? Cytochrome oxidase. In complex 4 of ETC, it's required that we have seen. Zinc for alcohol dehydrogenase, carbonic anhydrase. These are the example of major coenzymes and cofactors. From here, sure you will get one question. Okay. From here, sure you will get one question. Any doubts are there? Kindly let me know any doubts. Any doubts are there? Kindly reply, then only I will proceed things. Why today no interactions? Hello? Yes, no doubts. I know only 3 days is there. TK, don't take pressure. Just read and revise the things. TK. Now we are going to see the mode of enzyme. How the enzymes will catalyze the reactions. TK. How the enzyme will catalyze reaction. TK. From here they asked one time question. TK. The enzyme will catalyze the reaction by lowering the activation energy. This is very important. Okay. Enzyme will catalyze the reaction by lowering the activation energy. Okay. This is just for your information only. Enzyme will bind with substrate that will lead to formation of enzyme substrate complex. Okay. This enzyme substrate complex will cleave to form free enzyme and its product. Okay. This is just for your understanding. Okay. So if an enzyme is there, there should have one active site where the catalysis will happen. So what is meant by active site means it's a small cleft-like structure present in an enzyme where the substrate will bind and catalysis will be proceed. That is known as active site. This definition is important. Okay. So the enzymes are highly specific. This is another important thing. Enzymes are highly specific towards its substrate. Stereospecific will be there, group specificity will be there, absolute and bond specific will be there. Absolute specificity means what? Whether it will act on D or L form. Okay. Bond specific means which bond it is acting. Okay. So group specificity means which group. Okay. Stereo specificity means DL. Okay. Same DL will be there. Okay. So this is very important. Enzymes are stereo specific, bond, group and absolute specific. Okay. There are different theories are there to determine the enzyme activity. Okay. You just have to remember two theories are there. One is lock and key theory. It was proposed by M.L. Fisher. Okay. M.L. Fisher proposed this lock and key theory. Okay. So here what that person has told means the enzyme will have active site. The active site will be complementary to the substrate. So it will bind, then the catalysis will proceed. Okay. Then the disadvantage of this one is here that person has told this catalytic site is a rigid site. Okay. So it is perfect complementary like how a lock is complementary to its key. Okay. To overcome this, second theory came, that is induced fit theory or coastland model. Okay. Or coastland induced fit theory. Okay. Here the most important thing is this is most accepted theory, and the most important thing what he have discussed means the enzyme active site is what? That is not an that enzyme active site can make a conformation changes once the substrate will bind. Okay, this is the most important thing from this theory. Okay, then what are the different factors will affect the enzyme activity? This is just for your understanding only. Okay, you just go through this one. This is not that much important. Okay, now we are going to see the catalysis. Okay, enzyme kinetics and how the catalysis will happen. This is very important. Okay, from here sure you will get one question. Okay, in enzymology 2 questions you will get. From enzyme kinetics pakka you will get one question. Okay, so enzyme kinetics means what? If an enzyme is there, in which rate the substrate is converting to product. Okay, that is what? Kinetics. On which velocity the enzyme kinetics is happening. These are the different kinetic parameters. Okay, so enzyme kinetics means study of rate of enzyme catalyzed chemical reaction. Okay, this enzyme kinetics was given by whom? The equation was given by Michaelis and Menden. Michaelis and Menden. This is very important. Okay, Michaelis and Menden. The equation is what? V0 equal to Vmax of substrate by Km plus S. V0 equal to Vmax of substrate plus Km plus S. This is repeated question. Okay. So, velocity of the enzyme can be determined by V0 equal to Vmax of substrate by Km plus S. Okay. So, this is just a graph of V0 against S0. S0 is what? Substrate concentration. V0 is velocity of the enzyme. Okay. So, here coming the another term Km. Km means what? It's a Michaelis-Menten constant. Km means Michaelis-Menten constant. Okay. So, Km means what? Of the maximum velocity, the substrate concentration at which the enzyme showing of the maximum velocity. I hope this is clear. So, Km means the substrate concentration at which the enzyme showing of the maximum velocity. I am just going fast because these things we have discussed very nicely in our lecture. Okay. Now we are going to see about Line-Weaver-Burke plot. We know V0 equal to Vmax of substrate by Km plus S. This is Michaelis-Menten equation. Okay. So Line-Weaver-Burke plot is double reciprocal plot of Michaelis-Menten equation. So 1 by V0 equal to Km plus S by Vmax of S. Okay. If you derive the equation, we will get 1 by V0 equal to Km by Vmax into 1 by S plus 1 by Vmax. This is the double reciprocal plot. Okay. This we will call as Line-Weaver-Burke plot. Okay. Why we are going to do like this? Why we need this Line-Weaver-Burke plot? If you see, if you want to know the kinetic parameters, what is the maximum velocity of the enzyme, what is the Km of the enzyme, we can use this Line-Weaver-Burke plot to determine the kinetic parameters. Okay, this is just simple thing only. Okay, now we are going to discuss about enzyme inhibition. Okay, last topic, enzyme inhibition. Okay. Yes, enzyme inhibition. Okay. So why we have to study the enzyme inhibition? That question has to come to your mind. Okay. Why we are going to study the enzyme inhibition? So if you see in our day-to-day life, we are taking so much of drugs. Like for example, we are taking so much of tablets. Okay. The tablets will act as an inhibitor of specific enzyme in our body. Because of that, some benefit we are getting. For example, antibiotics we are using. This will act as an inhibitor for the bacterial growth in our body. So, why we have to study the enzyme inhibition means we can design the drugs that can targeting specific molecules in our body. Okay. So, these are the practical application. Okay. Now, we are going to see the enzyme inhibition. What is meant by inhibitor? What are the classification of enzyme inhibition? Okay. From enzyme inhibition, sure you will get. Okay. What are the classification of enzyme inhibition? These things we are going to see. Okay. So, enzyme inhibition means what? It's the process in which an inhibitor molecule bind to an enzyme and decrease or stop its catalytic activity. Okay. So, it's an inhibitor is a substance or a molecule that will go and bind with the active site of an enzyme. Then it will decrease the catalytic efficiency of an particular enzyme. The example are what? For example, antibiotics we have discussed. Okay. There are two broad classifications of enzyme inhibitors are there. Okay. For example, reversible inhibition and irreversible inhibition. This is two broad classification. Okay. Among them, the reversible inhibition are further classified into two types: competitive, non-competitive, and uncompetitive. This is the question area they are going to ask. Okay. In competitive inhibitor, how the Km and Vmax will change? This is the question they are going to ask. Okay. They will give in match the following. They will give what are the different inhibitors. Okay. And they will ask what are the kinetic parameters will be changed. Okay. So the key difference between reversible and irreversible inhibition is what? In reversible inhibition, the inhibitor bind to the enzyme in non-covalent bond formation. Okay. But in reversible inhibition, this will form a covalent bond with the enzyme. Okay. This is the key point you have to remember. Okay. Now we are coming to competitive inhibition. Okay. I already told the reversible inhibition is of three types: competitive, uncompetitive, and non-competitive. Okay. So competitive inhibition means what? The name itself it's there. That inhibitor will resembles the substrate. Okay. For example, this is enzyme substrate. This is inhibitor. This inhibitor will go on to compete with the active site. Okay. So it will go on to bind here, but it will lack the particular amino acids or group for catalysis. So the inhibitor will go on to bind with the enzyme. So catalysis will be decreased. I hope this is clear. So in competitive inhibition, the inhibitor will resembles the structure of substrate. It will go and bind in the active site. These are the two important things you have to remember. And most important thing, if I increase the substrate concentration, the inhibitor concentration will become less. So the catalysis or enzyme decreasing activity will also be less. Okay. So these are the key points. Inhibitor competed for the active site of the enzyme. It's a class of reversible enzyme classification. If excess substrate is there, means that will abolish the inhibition. In competitive inhibition, Vmax will decrease and Km will unchanged. This is the 2 point you have to remember. Okay. Vmax of the enzyme will decrease and Vmax will be unchanged and Km will increase. These are the 2 points you have to remember. Okay. This is not important. Okay. Then most important thing, the examples of competitive inhibitors. Okay. Malonate is an example for succinate dehydrogenase. Methoxyrate is an example for dihydrofolate reductase. Sulfonamides are example of folate synthase. Okay, these are the typical example for competitive inhibitors. Okay, if you want to see one most important thing in the previous slide, see the line viewer Berkey plot for competitive inhibition will look like this. Okay, they can ask in picture questions also. Okay, so see here only the Km value is changing, but Vmax value is unchanged. Okay. Only the Km value is changed here, but the Vmax is unchanging in competitive inhibitor. Okay. Now we are coming to uncompetitive inhibitors. Okay. So uncompetitive inhibitors means what? Inhibitors will bind particularly to the enzyme substrate complex. Okay. Inhibitor will bind to enzyme substrate complex. Okay. The most important thing what you have to remember here means the Vmax and Km both will decrease. Vmax and Km both will decrease. Vmax and Km both will decrease. Okay. Then another thing, okay, you have to just remember the Lineweaver Berkey plot, how the Lineweaver Berkey plot will resemble in uncompetitive inhibitor. Okay, it will be parallel lines will be there because both Km and Vmax is changing here. Okay. The next classification, non-competitive inhibitor. Okay, so non-competitive inhibitor examples. Okay, so from non-competitive inhibitor means the inhibitor can bind to the free enzyme, also to the enzyme inhibitor, enzyme substrate complex. Okay, so inhibitor will bind to different domains on the enzyme other than active site. Excess substrate does not abolish the inhibition, and the most important thing, Km will be unchanged and Vmax will reduce. Km will be unchanged and Vmax will be reduced. Okay. Safura, sure, we are going to take ASRB net biotechnology, biochemistry and physiology from this year onwards. Okay. We will update you. Okay. Then next thing, kinetics of competitive inhibition. This we have already discussed. Okay, I will show the brief difference between kinetic of different types of inhibition. This is very important slide. Okay, very important slide. Okay, you just remember this one. This is highly sufficient. In competitive inhibitor, the inhibitor binds to free enzyme, so Km will increase, Vmax will unchanged. Okay. In uncompetitive inhibitor, this inhibitor will bind to enzyme substrate complex, Km will decrease and Vmax also decrease. In non-competitive inhibitor will bind to free enzyme and also to enzyme substrate complex. So, Km will not change and Vmax will decrease. Km will not change and Vmax will decrease. I hope there is no doubt in enzymology. Any doubt is there, students? Kindly let me know. Any doubt is there? If no other doubt means we can end. Any doubt is there? Any doubt is there? I hope we have given our 100% for this revision classes. Okay. Still 3 days are there. Give your 100%. Okay. All the best for your examination. Okay. I will see you all in IRA. Okay. Thank you guys. Thank you for your listening. Thank you.